Everyone, this is Dr. Srikanth from Team MDS Conquer. Now I'm going to discuss few important topics related to the metabolism of carbohydrates. So I'm going to discuss about glycolysis. The first important point that you need to add is it's also called as EMP pathway. And the definition says glycolysis can be defined as a sequence of reactions converting the glucose or glycogen either into pyruvate or lactate. Pyruvate will be formed in the case of aerobic reaction in the presence of oxygen, and lactate is formed in the case of anaerobic reaction in the absence of oxygen. So there are some features that you have to learn about the glycolysis. Generally, this reaction takes place in the cells of the body, and the enzymes that are really required for this are present in cytosol. The second point is glycolysis can occur either in the presence of oxygen or in the absence of oxygen. In the presence of oxygen, it is called as aerobic reaction, and in the absence of oxygen, it is called as anaerobic reaction. So in the aerobic reaction, it results in the formation of pyruvate. In the anaerobic reaction, it results in the form of lactate. Third important point, glycolysis is a major pathway for production of ATP in tissues lacking mitochondria. Regularly, mitochondria is the one that is responsible for the production of ATP, and it is called as powerhouse of cell. But there are few cells where the mitochondria is lagging, in which glycolysis is the major pathway by which they generate energy. They are erythrocytes, cornea, and the lenses. Point number 4. Glycolysis is a very essential for brain, which depends upon glucose for their energy. So brain, the main important mechanism by which it get energy is glycolysis. And the next important point to be added, the fifth important point, is reversal of glycolysis along with the alternative arrangement of irreversible steps will resulting in the synthesis of glucose. That is nothing but gluconeogenesis. Means glycolysis is nothing but it's a reversal of gluconeogenesis with the arrangement of irreversible steps. We'll learn about what are reversible steps and what are irreversible steps. But if you provide an alteration for irreversible steps, the glycolysis process can be reversed, and the reverse process of glycolysis is called as glyconeogenesis. I hope you're clear until now. Now we are going to learn about the individual reactions. So basically the reactions can be categorized into one, that is energy investment. Means where you have to invest the energy. It is also called as priming stage, or also called as preparatory stage, where energy is utilized, invested. The second stage is called as splitting phase, and the third stage is called as energy generation phase, also called as payoff phase. So coming to the first one is energy investing, also called as priming stage, also called as preparatory phase. So what happens in this? Okay, so there are three major steps which occurs in this. One is the initial product, already known, already discussed, that the initial product for this is glucose. So glucose initially is going to convert into glucose 6-phosphate in the presence of either hexokinase or glucokinase enzyme. This already discussed, this is an energy investing phase where an energy is used. What is that energy? ATP is used. And the byproduct is ADP, and in the presence of magnesium ion. What is generated? Glucose 6-phosphate. So, as already discussed, you can see this is an irreversible step. Okay, this is an irreversible step. The points to be added about this is, so these hexokinase or glucokinase, both of these are isomers. Okay, these are isoenzymes. So what is the advantage of hexokinase? Like hexokinase is present in all the tissues. And it helps in catalyzing or helps in the reaction of hexoses. Hexokinase helps in the reaction of hexoses. The hexoses can be fructose or mannose or anything. And the hexokinase has low Km value. The value is somewhere around 0.1 millimoles. And this hexokinase is inhibited by the glucose 6-phosphatase. Means whenever this glucose 6-phosphatase increases, okay, the hexokinase activity will be inhibited. Coming to the second part, that is glucokinase. So glucokinase is mainly present in the liver, whereas hexokinase is present in all the tissues. Glucokinase mainly acts on glucose, whereas hexokinase can act on all hexoses. The Km value of glucokinase is more, and the value is 10 millimoles, whereas the Km value of hexokinase is low Km value. And the glucokinase is not inhibited by glucose 6-phosphate, whereas your hexokinase is inhibited by glucose 6-phosphate. So these are the important things that you have to make a note. So what is the advantage of having low Km value? So whenever you have a low Km value, which means that it has high affinity. So as we already discussed, that hexokinase has low Km value, means which has high affinity, means which can utilize the glucose or which can utilize the hexoses even when they are at very low concentration. Whereas when you're coming and talking about glucokinase, glucokinase acts only at higher concentration because its Km value is high. Means preferably glucokinase will come into action immediately after the meal, where the glucose levels will be more. But hexokinase can act even at lower levels because of its low Km value and high affinity. I hope you are clear with the first step, which is a very, 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 very important step. Why? You can ask me why it's important. Because the end product of the first step is glucose 6-phosphate. It is a central molecule for many metabolic reactions like glycolysis, glycogenesis, glyconeogenesis, pentose phosphate pathway. All these cycles or all these pathways, they have this as a prime molecule. So glucose 6-phosphate has lots of importance. So we are done with the first step. Now moving towards the second step. So what happens in the second step? Glucose 6-phosphate is now going to convert into fructose 6-phosphate. Means it is changing. Glucose is changing into fructose, and this reaction is mediated by an enzyme called as phosphohexoisomerase. The enzyme is phosphohexoisomerase. isomerase. I hope you're clear. This is an isomerase enzyme. Okay, so we are done. But make sure this reaction is a reversible reaction. This is a reversible reaction, whereas the previous reaction is an irreversible reaction. So we are done with the second step. So now moving towards the third step, where the fructose 6-phosphate is now going to convert it into fructose 1,6 bisphosphate in the presence of an enzyme called as phosphofructokinase. And even in this step, there is utilization of energy. Okay, phosphofructokinase utilizes energy where an ATP is used in the presence of magnesium to generate ADP. So I hope you are clear with the first three steps. The three steps are called as, what are they called as? They are called as energy investment. Means you are using some energy. Where you are using? First, glucose is going to convert it into glucose 6-phosphate by using one ATP molecule in the presence of hexokinase or glucokinase. Make a note about difference between hexokinase and glucokinase. Then glucose 6-phosphate is going to convert it into fructose 6-phosphate in the presence of an enzyme called as phosphohexoisomerase. Then fructose 6-phosphate is going to convert it into fructose 1,6 bisphosphate in the presence of phosphofructokinase by utilizing one more ATP. And this reaction is irreversible. This reaction is reversible. So we are done with the first three steps, which are called as energy investment steps, where you can see in 2 steps out of 3, we are investing energy. We are investing the ATP molecule. So the second phase is called as splitting phase. Means you're going to split something. What you are going to split? This fructose 1,6 bisphosphate is going to break into 2 parts in the presence of an enzyme called as aldolase. It's going to break into dihydroxy acetone phosphate, and the other molecule is glyceraldehyde 3-phosphate. So fructose 1,6 phosphate is going to split in the presence of split. So that is the reason why it is called as split phase. It's split into 2 molecules. One molecule is dihydroxyacetone phosphate and second molecule is glyceraldehyde 3-phosphate in the presence of aldolase enzyme. So this is called as splitting phase. Clear? Now what is going to happen? Now dihydroxyacetone is going to convert into glyceraldehyde 3-phosphate in the presence of phosphotriose isomerase. So previously we have one molecule of glucose, glyceraldehyde 3-phosphate. Now we have another molecule. So now we have 2 molecules, because this is going to convert it into. So we have 2 molecules of glyceraldehyde 3-phosphate for now. Okay. So with this, we are done with splitting phase also. We are done with energy investing phase with 3 reactions. We are done with the splitting phase with 2 reactions. And the end product of splitting phase is 2 molecules of glyceraldehyde 3-phosphate from 1 molecule of glucose. We have initially started our journey with 1 molecule of glucose. Now the end product of splitting phase is 2 molecules of glyceraldehyde 3-phosphate. So now what is going to happen? So we are done with investment. We are done with splitting. We are done. Now the end product is energy generation. Okay. So this is how your preparation will be. I can just compare these phases with your preparation. The first phase is investing phase. Invest your time, invest your hard work and everything. The second phase is splitting and reading. Divide and read, divide and revision. And the last phase is energy generation, means success phase. So once you're done with the exam and everything, the next half will be the success phase. So now the phase is energy generation phase. So what we are left with? We are left with 2 molecules of glyceraldehyde 3-phosphate. Okay. So what is going to happen? So first, initially, so this glyceraldehyde 3-phosphate is going to convert into 1,3 is going to convert it into 1,3 bisphosphoglycerate in the presence of an enzyme called as glyceraldehyde 3-phosphate dehydrogenase. So in the presence of glyceraldehyde 3-phosphate dehydrogenate, 3 glyceraldehyde phosphate is going to convert into 1,3 bisphosphoglycerate, in which NADH plus H plus is generated. I hope you're clear with this concept. Okay, so we have started our 6th reaction. This is a 6th reaction where the glyceraldehyde 3-phosphate dehydrogenase is going to help in converting the glyceraldehyde 3-phosphate to 1,3 bisphosphoglycerate. And this step is important as it is involved in the production of NADPH plus, a high energy compound that is 1,3 bisphosphoglycerate. And of course, this NADPH passes through electron transportation chain and helps in generation of ATP, which we will be learning. So this is one of the oxidative phosphorylation level. Next one, so this is going to convert into the next level. So we are done with the 6th reaction. Now we are going with the 7th reaction. The 7th reaction is nothing but conversion of 1,3 bisphosphoglycerate to 3-phosphoglycerate in the presence of phosphoglycerate kinase. Phosphoglycerokinase. And this is a place where there is generation of ATP. Until now we have utilized the ATP, and this is a phase where ATP is generated, and this level is called as substrate level phosphorylation. So the very unique properties of this 7th phase, the 7th reaction, is this is called as substrate level phosphorylation, and this is the only kinase enzyme which is reversible. Generally, if you see, go back and just have a look about your kinases. Okay, this is the first kinase which is an irreversible reaction. This is another kinase which is an irreversible reaction, and this is the third kinase enzyme, but it's a reversible reaction. Make a note. This is the phosphoglycerate kinase is the only kinase which is a reversible reaction, and this reaction is a reaction where there is generation of ATP for the first time, and this reaction is called as substrate level phosphorylation. So right now you have 3-phosphoglycerate in your hand, which is going to convert into 2-phosphoglycerate. In the presence of an enzyme called as mutase, that is phosphoglycerate mutase. Then this 2-phosphoglycerate is going to convert into phosphoenol pyruvate in the enzyme called as enolase. Okay, so this is very important for dentistry because this is an enzyme which is activated, or which is inhibited by fluoride. So when you use fluoride, when you add fluoride, so what is going to happen is the fluoride is going to form complexes with this magnesium, and it's going to inhibit or decrease the magnesium levels, by which it's going to inhibit the enolase enzyme, and by which it's going to decrease the production of ATP for the bacteria, by which it decreases the bacterial count. And this is the same reaction, or this is the same mechanism by which the exact glucose level values of a patient can be determined in blood banks. Okay, I hope you're clear about this. So we are done. So we got phosphophenol pyruvate. So phosphophenol pyruvate is going to convert it into pyruvate, where there will be generation of again an ATP. Okay, so the enzyme pyruvate kinase is the one which helps in the catalysis of this, and this is again generation of ATP called as substrate level phosphorylation. So here we have two substrate level phosphorylation. One is this one. This is the one substrate level phosphorylation, and the second substrate level phosphorylation is this. So you have two substrate level phosphorylations by which the pyruvate is generated at the end. This pyruvate generation is called as aerobic method. There is other method called as in the absence of oxygen, that is anaerobic, where there will be production of lactate. Hope everything is clear as of now. Okay, the next thing to be added is about how to count the number of ATP that are generated. So now I'm going to discuss about the recent modification. According to the recent modifications, I'm going to talk. Okay, there are few textbooks or few MCQ books still going with the old method, and whatever method I'm talking is a recent one, and it is according to the 5th edition of Sachin Narayana, which is the latest biochemistry book available in the market now. So coming to glycolysis. So in glycolysis, you have a step where you're going to have an ADP. This is the one. But what is going to happen? As you have two molecules, you'll have two of this. So you have 2 NADH. You have 2 NADH. So according to the recent modification, 1 NADH is going to give rise to 2.5 ATP. So 2 NADH is going to give 5 ATP. Hope you're clear. And the next one, you have two substrate level phosphorylation where you are generating the ATP, right? One is this, where you have an ATP. But we are talking about two molecules, because we have two molecules of glyceraldehyde 3-phosphate. So we will get 2 ATP. So here 5, now it's 2, now it's 7. Again, in the last step, you have 2 more ATP. So you have totally 9 ATP are generated, of which we are just counting the ATP which are generated in the generation phase. But during the investment phase, we have used 2 ATP. See here, we have used 1 ATP here, we have used 1 ATP here. Totally we got 9 minus 2, so it is 7 ATP. I hope you are very clear about the number of ATP generated during the net number of ATP generated, 9 minus 2. So the net ATP generated in the glycolysis in the aerobic is 7 ATP. I hope you are very clear about this. And for example, when a glycolysis occurs from glycogen, here the glycolysis occur from glucose. For example, if it is occurring from glycogen, one more extra ATP is generated. Why one more extra ATP is generated? Because there is no need of this conversion, because there is no need of utilization of the first ATP, which is just used to activate glucose 6-phosphate, glucose to glucose 6-phosphate. So there will be addition of one more ATP. Thus, in anaerobic glycolysis, 3 ATP are generated from glycogen. That is more advantage when compared with the glucose. I hope you're very clear about this. And few more application things that to be added is about the cancer cells and the glycolysis. The cancer cells, cancer is uncontrolled division of cells, right? The simple basic definition. The cancer cells displays an increasing in uptake of glucose, and there will be increasing in the activity of glycolysis. As the tumor grows rapidly, the blood vessels are unable to have adequate oxygen, so the tumor grows very rapidly. The oxygen levels to the blood will drop down and lead to hypoxia. So what is going to happen? The aerobic cycle is going to convert it into anaerobic cycle. And this hypoxic inducible transforming factor is a factor which is present in these cells, will activate and increase the synthesis of the enzymes that are responsible for glycolysis, as well as they helps in transportation of glucose. However, the cancer cells cannot grow and survive without proper vascularization. One of the modality of the cancer treatment is to use a drugs that is going to inhibit the vascular supply to the tumor, by which the cancer can be cured. So apart from this, you can make a note about the irreversible steps in glycolysis. We have already learned. Okay, first if you see the first phase, that is the energy investment phase, there are two irreversible. One is your kinase, hexokinase or glucokinase is the first irreversible step, and the second one is phosphofructokinase is the second irreversible step, and going to the last one, that is this is the pyruvate kinase is the third irreversible steps. So the easy way to remember all irreversible steps in glycolysis are kinases. There are 4 kinases, out of which one is a reversible kinase. We have learned that PGK is a reversible kinase, you can exclude that, and rest all are irreversible steps. And the basic difference between the glycolysis and glyconeogenesis is just the irreversible steps. So you need means it's going to be reversal. Okay, it's going to be the reversal. So you need to give an additional enzymes for all the irreversible steps, means all the 3 irreversible steps you need to have an alternative, so that with that alternative the entire process can be reversed. So for example, if you are planning to go back pyruvate to this one, this is a single directional reaction, so you need an extra enzyme for it. And from here to here is a reversible, it will go back. Here to here is a reversible, it will go back. Here to here, this is the only kinase which is a reversible, it will go back. This will go back. This is a reversible reaction, it will go back. It will go back. Here we need an extra enzyme because it's an irreversible reaction. It will go back because it's a reversible reaction. It will not go. We need to provide an extra sum. So that's Just an alteration of the irreversible reactions will make the cycle reverse, and that reverse cycle is called as gluconeogenesis. Done for now. We'll be coming with few more videos to help you out. The only thing that we can follow now is a simple principle. Try to be positive. Learn as much as you can. Okay, so biochemistry is something called as dry topic, but if you take the rough analysis of NEET exam or any other exam into consideration, biochemistry carry half of the weightage of a regular subject. Means it's going to carry 7 marks. Regular subjects are going to carry 14. Similar to that of AIMS, clinicals will carry more marks, and coming to comparison with the clinical, biochemistry is going to carry less than 50% of marks compared to any other subject. For example, if you're taking ortho as a subject, so if you just compare the weightage of ortho to that of the biochemistry, either in the AIMS or in the NEET, biochemistry is going to carry less than 50% of marks when compared with ortho. So don't get panic. The only suggestion what I can give you is just try to be the best, whatever it is, whether it's biochemistry or ortho. Try to be the best, learn as much as you can, try to be positive. Okay, don't divide or analyze the subjects basing upon their weightage. But now you are in a phase. What phase? Already discussed. You are in energy investing phase. Invest as much as time as possible, as much as energy as possible, as much as hard work as possible. Then you will have a great return. Similarly, like your glycolysis, where your energy generation phase will be there. You're going to generate something great with whatever you're investing now. Thank you for now. Signing off, Dr. Srikanth from Team MDS.